Hi guys. How are we doing? Good afternoon, good afternoon. Okay, so I want to jump on here because obviously I'm going to try and do as much as I can for you guys as we are going. But one thing I would say, I don't know why, but in my gut, I have a feeling that there's going to be something on gene regulation. I've been trying really hard to get my revision guide done for you, but I haven't finished it. So I want to go through the content of gene regulation and how that is involved in what you need for moving forward. So let me just so most important thing to begin with is noting that you should know, linking to module 2, that you have got DNA, and in that DNA, here we go, just make this a bit bigger. You've got nucleic acids, so nucleotides, and those nucleotides are very much, 1 second, just going to make this a bit bigger. There we go. DNA is made up of genes, and those genes are made up of nucleic acid, and that nucleic acid is going to be transcribed. So the important thing is that I have got 3 different genes here, gene 1, gene 2, gene 3. And what needs to happen first is transcription and translation. What topic is this? So I'm looking at gene control, and this is module 6, gene control, because I feel like there's not been that much on regulation of genes. So I feel like they could do some parts on gene regulation. And a lot of you have said that module 2, DNA, and the transcription translation didn't come through very much. So what I would say is this is where it can all interlink together. So you should know that you have your DNA. Your DNA needs to be transcribed onto my mRNA. But remember, RNA molecules are not just mRNA. RNA could also be talking about our tRNA and also talking about our ribosomal RNA. So if we're looking at module 2 and you are talking about 2.1.1 cell structure, you should know that inside cells there are ribosomes. And those ribosomes, depending if we're looking at a prokaryotic cell or whether we're looking at a eukaryotic cell, those ribosomes could be 70S or 80S, but what's going to happen is translation will occur at the ribosome. And to be able to do translation, we need both mRNA and our tRNA. But our ribosomal RNA is needed to actually make the ribosomes themselves. So ribosomes, you know, are made of protein and ribosomal RNA. So make sure you don't get hung up on the fact that transcription is just mRNA, because they may throw in a curveball and talk about just polynucleotides. Excuse my writing as I try and do this really fast for you. Is a lac operon in the nucleus? No, Tony. The lac operon is prokaryotic cells. So remember, the lac operon is talking about the E. coli. So therefore, we know it's not in the nucleus because we know our prokaryotes have got a DNA that is, oh my word, my writing today is atrocious. I'm trying to do things so quickly. Do you remember what I said to you? If you get that feeling that you're going too fast, just breathe it out. So prokaryotes, we know our DNA is a circular DNA, and we know that it is not contained in a nucleus. So we know that Our prokaryotes have DNA that's circular, not containing the nucleus, but we know that they need translation to occur, and therefore they need ribosomes. So if they were talking about biotechnology and they were talking about bacterial cells, yes, this is OCR, guys. I'm only focusing on OCR, and that's what my content is for, is OCRA as well. But just to go back here, the proteins that are going to be produced, whether we're talking about prokaryotes or whether we're talking about eukaryotes, those proteins are made up of amino acid sequences. And those amino acid sequences are important because if we change the amino acid sequence, that could have an effect on the structure of the protein. So whether we're talking about proteins within ribosomes or whether we're talking about proteins within any other type of structural gene, that is what we're looking at. Keywords to come through: amino acid sequence, which is then going to determine your protein's primary structure. And that protein primary structure will then determine the alpha and beta helixes, or alpha helix, beta plated sheets, and then your quaternary structure. So it's really important when you're doing this topic area, and Tony, I will cover the lac operon because it hasn't come up in a while. Can mRNA bind to any ribosome or only specific ones? So mRNA binds to the small subunit of the ribosome. So it's going to bind always to the small subunit of the ribosome, and it binds at a very specific region. So the fact that your mRNA always has what's known as a start codon, and that start codon actually codes for methionine. So every single polypeptide starts with the same amino acid, methionine, and then depending on how it is actually modified as to whether that methionine is removed or not. So the mRNA is going to bind to the small subunit, which is, hold on, let me just go back to that one. Too many things open at the moment. Just close this all down. There we go. This is the small subunit. So this one is the small subunit, although it looks big. And this one is the large subunit. Do you need to specify that it binds? No, you just say it binds to the ribosome. But it's important to understand how the process actually works sometimes, because then it helps you. If you go into making it too basic, it can make it a bit confusing, but they won't expect you to state that in the exam. Do you have to remember the name of the start amino acid? You should really know the starting amino acid is methionine, because that's your start codon. That is like a knowledge that you should have, because every single one starts with methionine. So knowing that you have, and I don't think they expect you to memorize the amino acid as being the start, the methionine is the start amino acid, but you should know that there's a start codon. So if they're going to expect anything, it would be the start codon as being the important one. So a start codon. Now, important to note when we're talking about, and I'm going to bring in the lac operon, I've been updating my diagrams. If we're looking at the lac operon, then it's important to note, just see if I can copy this across. Oh, 1 second. Equal it. I can make this, I don't know if I'll be able to make this bigger. That makes sense. Try copy it straight across from my bit clip. Try that again. Let's go and zoom in. Okay, so what do we have then? When we look at the lac operon, a lac operon, so when we talk about prokaryotic cells, they have an operon, and that operon is where you have a cluster of genes, which are all under the control of the promoter region. So here is my promoter region. So I've got two sections here that I'm showing you is my promoter region. Now, the promoter region is always just slightly upstream from where the genes are. So as you can see here, I've got three structural genes if I was looking at the lac operon. And so therefore, our RNA polymerase, we know that RNA polymerase is about building our phosphodiester bonds. And it builds the phosphodiester bonds on the adjacent mRNA nucleotides, because remember, they are nucleotides. They are made up of a phosphate, which is attached to a ribose sugar and a nitrogenous base. So it's important that you are trying to go back and remind yourself about these key terms, because Module 6 is just putting Module 2 into context. You have got this promoter region, RNA polymerase is going to bind there. But what's really interesting is when RNA polymerase binds to the promoter region, it actually activates the helicase to do the unzipping. So if the RNA polymerase cannot bind to the promoter region, there's helicase is not activated to unzip the double helix strand, and therefore we're not exposing our template strand. Because remember, when we talk about transcription, we have a template strand, which is going to be transcribed. So it is important when you are looking and revising the content that you do think about those key terms from Module 2. So what we have is the operator is going to block the RNA polymerase from binding if there is a repressor protein attached to it. So you need to know that we have some regulatory genes, and when I say we, I'm talking about the prokaryotes here. They have these regulatory genes which are going to encode for a repressor protein. Eukaryotes can also have regulatory genes that code for repressor proteins, but prokaryotes are more simplified because they just have a simple, say it's simple, but not really, circular, not associated with histone proteins. Their circular DNA doesn't have any histone proteins attached to it. And therefore, because they don't have any histone proteins attached to it, that's going to mean that they are not actually going to need the complicated side of things of transcription factors. So what I want to do is, maybe I will just do, I'll give you a little sneak peek. She says it's not finished yet, it's in the process. What's the role of transcription factors in this? Okay, not in this one. So hold on, I'm going to get you down to this one here. So when we look at transcription, we know transcription happens in eukaryotes, but what I'm interested in looking at right now is transcription in prokaryotes. So when we talk about the lac operon, when we talk about the group of genes under the control of a single promoter, we're talking about them being transcribed together. So what you can see here with the lac operon is you have got the no lactose in the environment. Well, we still are going to transcribe and therefore create our mRNA. The mRNA is encoding for a repressor protein. If there's no lactose in the environment, the repressor protein is going to bind to the operator. And because it's binding to the operator, it is blocking the RNA polymerase from being able to bind. If the RNA polymerase cannot bind, it cannot transcribe these three structural genes. If it can't transcribe these three structural genes, it's therefore not going to be doing translation of the mRNA. It's not going to be wasting any energy in that whole process. So the reason the E. coli actually do this is a form of conserving energy. Their respiratory substrate of choice would be glucose. Now, we know that lactose is a disaccharide that's made up of glucose and galactose held together by a glycosidic bond. Therefore, we need to hydrolyze it down to produce the glucose and galactose that can then be used as the respiratory substrate of choice. So when lactose is present in the environment and the glucose concentration is low, so this is where the environment has an effect, the environment is going to actually cause for the regulation of the gene expression. So we've still got mRNA being made, we've still got transcription, we've still got a repressor protein, but this time our lactose, our disaccharide, is binding to the repressor protein. Look at that change in the shape. The shape has changed here, and because the shape has changed, it's no longer complementary, so it cannot bind to the operator. That therefore means that the RNA polymerase is now free to bind to the promoter, and when it binds, that means transcription can occur. And therefore, what you end up having is that the transcription that's occurring, excuse me, the mRNA of all three structural genes will be made as one, okay? So the RNA polymerase is going to make its way down, building the phosphodiester bonds. And because it's building the phosphodiester bonds, that's therefore going to create this long structure. So a transcription factor, Grace, is something that's going to either initiate or is going to inhibit the process of transcription occurring. So in this case, the lactose is actually called an inducer. If the lactose is present, it is going to induce the transcription of those three structural genes. And so therefore the repressor protein inhibits it. So be careful when we talk about transcription factors, we tend to talk about them more looking at the eukaryotic organisms. So when we're doing eukaryotic organisms, we are talking about the fact that we are either going to activate, promote RNA polymerase from binding, or they can be repressors where they're going to block the RNA polymerase from binding. Excuse me. So it really just depends where they go with the question as to what they're going to ask you on that front. Does that help? Let me go back here. So this is one thing I do feel like they're going to potentially go down the route of with the transcription factors, with the what am I trying to say here? Transcription factors or the lac operon. I feel like gene regulation is absolutely coming through. But obviously, you've also got with gene control, you've also got the fact that you have your mutations that could occur. So they could look at mutation is the operon and non-coding gene. Do you mean the operator? Be careful there, the matrix. The operon is the whole structure that we're talking about. It is the structural genes which are actually connected together and under the same promoter region. So the operator where the repressor protein binds to, that is a non-coding gene. Yes. Yes, this is OCR. What would try that again? Would we have to define regulator gene? They may actually ask you to define a regulator gene. Yes, so you're talking about a gene that's going to code for something that's regulating the transcription of another of the operon. So the key thing is if they're asking you to define the regulator gene, the regulator gene is the gene that codes for the repressor protein, which will have an effect or by binding to the operator. Yeah, that's all right. Thing is, sometimes we can make mistakes with our writing ability, and when we're speaking, you'll hear it. I'll make some mistakes as I go. The difference is I'm okay with making those mistakes, so I know by verbalizing it, I'm listening to myself and picking up any flaws that I'm making. Is this love going to be recorded? Yes, this love is recorded. Yeah, the live is recorded. And it should be staying on my YouTube channel for you to watch again. Yeah, Tony, it's just the lac operon is an operon. Basically, it is a section of the gene. So if we think about it, right? So here we have, going back very basic here, here is my circular DNA. So there is my circular DNA, and on my circular DNA, I have got, I can't remember the colors I used. So I've got a green section, which was my regulatory gene. And then I have my yellow section, which was my promoter. I had what kind of brownie color? I don't really know. I literally was trying to do it really quick earlier. Just put that there. So here's my operator. Here is my promoter. And then I have got my structural genes following after. I don't remember what colors I've used, so I'm just making this up as I go. It's purple. So what you have then is your structural genes. Structural genes are here. 1, 2, 3. So that's my Z, my Y and A. Here's my regulatory gene, which is my LAC-I. And therefore, that is a section on the circular chromosome within the prokaryotic cell. What other parts of the operon are non-coding genes? What other parts of the operon are non-coding genes? Why are you asking that question with regards to, you thinking about whether or not you were going to splice out the introns, the non-coding regions? What are you trying to figure out with that part? The most important part is that you can name what they do rather than fixating on what a non-coding gene is. A non-coding gene is one that doesn't code for a functional protein. They're not going to ask you for more detail than that. Potentially, do I think the role of apoptosis would come up? Potentially, it could come up. Addy, we're not interested in paper one. Sorry, this is paper two. Let's move on. Paper one's done. Go and look at the comments that are on my previous videos if you're really interested. But it's really important you focus now on paper 2 and 3 because you can do nothing about paper 1 anymore. It's just the opposite of mitosis, essentially. So it's an interesting one. Abigail, apoptosis is not the opposite of mitosis. What's really interesting, apoptosis is about programmed cell death. You don't want to activate your immune response. You want to control the death of the cells without causing inflammation. So it's a really important way in which we are actually going to control programmed cell death, control the death of cells in a way to reduce the inflammation that could be caused, because inflammation and too much buildup of fluids could cause a problem. So you want to do it in a very specific way, and that's where apoptosis comes in. Apoptosis is really important as well when you're looking at, for example, the digits on my fingers. So when you start to develop as a fetus, we all go through the same process of development because of some conserved genes or homeobox genes. So we all have our heads at the top, our tails at the bottom. We have the fact that our limbs should come out in specific areas. So when you look at the development, what will happen is the cell mitosis will cause you to have more cells. So I've got more cells, more cells, more cells, more cells, more cells. But right now, my cells are all connected. And only when apoptosis occurs does it then sculpt it into having the separation of the digits. So what you need to understand is you need mitosis for the production of more cells through the process of our chromosomes. DNA replication takes our chromosomes to being chromosomes made up of sister chromatids, and then we end up with two daughter cells at the end of that nuclear division. Through the process of mitosis, we get our two daughter cells. So we're making many copies, lots of cells which are genetically identical. The apoptosis is what sculpts it into having the fact that you have obviously your separated digits, for example. So it's the two coming together, mitotic division making more cells. The apoptosis is about ensuring that things are in the right positions where they should be and functioning the way that they should do. Cloning, biotechnology, and manipulating genomes in this live, please. I am going to be going live over the next week leading up to Friday. I will be doing different lessons on different things. So I will bring in different parts. But right now, I want to focus on cellular control and answer any questions with regards to that. Do we need to know how the structural genes are lac Z and lac Y? No, what they may do is they may give you that information in the exam, and what they're expecting you to note is if we're looking at the lac operon, that we're talking about our lactose, as I said before, is a disaccharide. So this is where they could actually bring in biological membranes and ask you about the fact that you have a disaccharide. How is it going to get across that phospholipid bilayer? Because it's a big molecule, it's also a polar molecule. So how is it going to get across? So the only way it can get across is if you obviously have a protein which allows that to get across. And that's what one of those structural genes is about. It's about producing the protein that will allow for the lactose to be moved across into the cytoplasm so it can then be hydrolyzed. Well, again, we need to hydrolyze it. So therefore, we also need to produce the protein that is going to hydrolyze the lactose down into the monomers. So how are we going to ensure that they're broken down into the monomers? Well, we do that through potentially, they may look at reducing sugar test. I don't think anyone mentioned a colorimeter coming up in paper 1. So if they haven't brought in colorimeter, I haven't seen the paper, so I don't know. They could bring in colorimetry here. Why do introns need to be spliced out? Well, the introns that are spliced out, those introns are actually potentially useful elsewhere. So those introns could be useful in making the tRNA molecules. Those introns need to be spliced up so that they can be used for making ribosomal RNA. It just depends on what the role is, but also by doing the splicing and the process of splicing, what you're actually getting is that the exons, if I have exon 1, exon 2, exon 3, exon 4, and exon 5, what will happen is when I splice out and I join together my exons, I can end up with all of those exons being together in that order. Or I might splice it and end up with exon 1, with exon 4, and then exon 5. Well, what have I got? I've got a different sequence of nucleotides. That sequence of nucleotides is read in a triplet non-overlapping code, and therefore that will code for a different sequence of amino acids and therefore producing a different protein. So, for example, you have long chain and short chain insulin molecules that can be produced depending on what is required. Does apoptosis have to do with the G1 checkpoint? So apoptosis, if we're talking about apoptosis, that cell death, that wouldn't be involved in your G1 checkpoint unless, are you talking about the fact that the cell is needing to be destroyed? Because the G1 checkpoint is where you're checking that the cell has grown to the right level, that the cell has the right parts to it that it requires. So like the right enzymes, so protein synthesis has been occurring, it's got the right number of organelles, that there's no damage to the DNA. And if there is damage to the DNA, then it could result in apoptosis. If that damage is not repairable, then yes, that could be involved, but it may not just be that, because remember, after the G1, some cells will go into G0, and that will result in them in that resting phase. Yes. So is the location of where apoptosis occurs. So when you're talking about the location of apoptosis, we're talking about apoptosis being this programmed cell death. We have got genes which are going to initiate apoptosis and genes which are going to inhibit apoptosis. So it's important that when we're looking at this, let me just throw in a previous one. So if we're looking at, for example, what have we got here? So we know mitosis is about producing these genetically identical daughter cells. We know that we've got control of that by various genes. So you've got your proto-oncogenes, you've got your tumor suppressor genes, which are to reduce cell division, to repair the damaged DNA, and promote apoptosis. So you have different genes based on what is required by the cell and what it needs to do. So when we talk about apoptosis and we talk about the programmed cell death, the cell just shrinks, the nucleus condenses, it will then pull in its membrane, and this creates vesicles, membrane-bound apoptotic bodies, they're called. And basically that means that the phagocytes can come along and they can engulf them, and then you're not wasting cellular particles that could be then used again. Would that protein letting the disaccharide through the phospholipid bilayer be a channel or a carrier protein? It depends. To be honest, in the exam, they won't penalize you for saying it's a channel or carrier protein as long as you are saying it's a transport protein. The important thing is if it is a channel protein, it's moving it by facilitated diffusion. If it's a carrier protein, it's also moving it by facilitated diffusion. It's just about whether it is needing to just be a channel that opens, or whether it is one that does a bit of a flip-flop because it's a carrier. What happens once the phagocyte has engulfed it, what it will then do is it will then use its lysozymes to digest the material down, which now puts them back into their smaller subunits that can then be used again. So if you think about how do we do DNA replication? DNA replication is where you have free nucleotides which are then going to be used to build, to make the new strand. Well, where does that come from? That has to come from the materials that are within the cell. So it's about thinking about it like a factory that's having to constantly build something, break it down, rebuild it, break it down, rebuild it. So any type of metabolic reaction that's occurring is going to require energy. So for the breaking down and the building up, you are going to lower activation energy by having the presence of enzymes there. So therefore, that's going to help the process. If we want to build things back up again, we need enzymes. So it's all, in a sense, interlinked. So as long as you understand that those free nucleotides are required to do the building, or those free amino acids are required to do the building, then that's what's important. And then the other thing, as I said before, with regard to the programmed cell death, the cell shrinkage, the chromatin condensing, the membrane-bound organelles, that's ensuring that you are preventing any potential damage or inflammation. You don't want to damage the body. You don't want to cause inflammation, because if you flood an area with water, that can actually become a problem, because what you're then doing is you are changing the osmotic potential of that area. Therefore, that could actually cause some of the cells to swell. If it causes some of the cells to swell, they could end up rupturing, which becomes a problem. So inflammation is a good thing, because it will therefore prevent the spread of a pathogen. But if this was a process in which we are programming it, we want it to occur, then we don't want that to happen and cause inflammation. We're actually doing it because we need it, not because there's a pathogen present. Why can't the podocytes divide? The podocytes can't divide because they've already differentiated. So when a cell has differentiated, when it has got a very specific role to play, like the podocyte, it has a structure. That question was about the asymmetry, that is now asymmetrical. And so because of that, because it's specialized, because it's already switched off genes, gene regulation, it has done that at the transcriptional level. It has switched off the genes, but when it switched off the genes, it switched them off permanently. So this is why you have to be aware that the differentiation part of cell division, when cells become differentiated, what we're doing is we are switching off the genes, and that could be a permanent switch off. It's never going to switch back on again. Or it could be that they're switched off temporarily until there's an environmental signal that means they have to be switched back on. So that's where you have that transcriptional level modification, those transcription factors. We've switched them off for a time period, but if there is a hormone or an environmental condition that results in them needing to be switched back on again, that will then cause the transcription factor to be activated, and that will then allow for transcription to occur. So this is where, for some of you, you'll think to yourself, oh, well, hormonal system was only for paper one. Well, actually, it's not. If you understand that hormones are chemical messengers which can then activate transcription factors, they could be assessing your module 6 content, but still related to module 5 if they're looking at a hormone, for example. But they may not, because they may do it down the lac operon and talk about the environmental signal being lactose, which is module 2 because it's a disaccharide. Yes, good to hear. That's a good answer there. And doesn't have the ability, yeah, if it can't to go through mitotic division, if it can't produce the spindle fibers to, one, it can't replicate its DNA. Two, it also can't then separate them to do that division. And it's important to remember the cytoskeleton is huge here. If we're talking about mitosis, we're talking about the need of the cytoskeleton to move those chromosomes to the position, to then pull those sister chromatids apart, and then to move and pull the membrane for that division during cytokinesis. All of that involves the cytoskeleton. So all of that is energy demanding processes. So you don't want every cell to have the capability of going through mitotic division because then you'll end up with too much. So it's all down to signals which are to do with concentrations, which are linked to your cyclins, which you don't need to know about the cyclins, but you do need to know that they are, in a sense, activated by concentrations. And again, if they ask questions on that, it will be very much a application where they've given you some information. Sahib, I did say if you want to look at how people did on paper one, go and have a look at the comments that we made on my previous videos. I want to try and make sure that these sessions are very much about content moving forward. You can't do anything about paper one, so don't stress about that one. Amna, I'll probably go through papers like I do normally in my evening lives. I'm very aware I've got to go pick up the girls in like 10 minutes. So I want to just throw out a video when I can and do these lives when I can. Are there any more? Everyone's jumping on that one. How do I think it looked? I don't know. I haven't seen it. I won't actually get to see it because I'm not inside in the school, so I won't get to see it until I don't actually know when I'll get to see it. If Monday or Tuesday potentially, if I manage to get a copy of it, but there's no guarantee. So basically I am using what everyone's told me to help guide me. Can I explain the Hox genes? Of course I can. That'll be the last thing I finish on because this is very much linked to this topic area. So if we're looking at the Hox genes, what's really important when we do the Hox genes is that you know that it's a gene, okay? So to start with, we're talking about genes. We're talking about genes that have got a very specific role to play. So when we talk about the anatomical structure in eukaryotic organisms, we are talking about genes which are controlling the position where things actually go. So when you look at the starting stage of the embryonic stages within organisms, you'll see that they follow the same sort of pattern, and then they develop into more complex organisms depending on what organism you're actually looking at. So if we're looking at Drosophila, obviously very small, they are these fruit flies, they've only got 4 chromosomes, whereas if we're looking at humans, we've got 46 pairs of chromosomes. We have so many more genes, so therefore we're more complex. So there's a lot more involved when we look at the humans compared to Drosophila, but they're both animals. They're made up of animal cells. Therefore, they have the same types of cells. It's just about the information that they've been given because of the number of genes that they actually contain within their nucleus. So again, that's linking it to the fact that you've got eukaryotic cells. The DNA is associated with histone proteins. The fact that it's contained within a nucleus could be where they go down the route of, but specifically the homeobox genes, they are basically a gene which is going to determine the development of the embryonic stages. So the head at the top, the tail at the bottom. So if you look at, for example, insects are segmented organisms. They have 3 segments to them. They have a head, they've got a thorax, they've got an abdomen, and the head is always where it should be. The antennae come out where it should come out. They have 3 pairs of legs which come out at, say, the same position, and therefore it's about how these are positioned in the body based on the genes. So highly conserved section of DNA, highly conserved section of DNA, we have no mutations or very small mutations occurring within this section, this homeobox gene. So the homeobox itself is 180 base pairs. So 180 base pairs, that is therefore encoding for 60 amino acids because we know that 3 of those DNA bases code for 1 amino acid. So if we've got 180 base pairs, We divide that by 3, we get about 60 amino acids. So these 60 amino acids are the same, okay? Now, they are similar in animals, they are similar in plants, they are similar in fungi. If there are mutations that occur in the homeobox, that can result in, for example, legs coming out of where the antennae should be. So if there is any disruption to the homeobox, that can have an effect on the development of the embryo going through the stages. So there are drugs like thalidomide that was given as a morning sickness pill, and that resulted in confusing the signals as to where the hands and the feet should start to develop. And so you basically have a blueprint to you to say your head is here, your tail is here, your fingertips should be out here, your feet and your toes should be out there. And therefore, if anybody makes a mistake in that blueprint, i.e. there's any type of mutation that occurs, that's going to result in the wrong messages being relayed and therefore the development being affected. And so it's all to do with the fact that these are transcription factors. So the homeobox genes are coding for these transcription factors, these proteins that are going to bind to DNA, activating them, and therefore either activating or deactivating. Are the Hox genes and homeobox genes the same? The Hox genes are specifically in animals. So the homeobox genes within the homeobox genes is a homeobox section, 180, that codes for transcription factors. So a Hox gene is only found in animals. The homeobox is found in animals, plants, and fungi. The Hox genes only found in animals. And therefore, the Hox genes are the ones that are responsible for the development of the body plan in animals. So when we talk about plants, we're not saying they've got a head and a tail. They have the apical meristem at the tip of the root and the tip of the shoot, other way around. But if we're talking about the top of a plant, that's what we're talking about, the apical meristem. They are controlled by the homeobox genes within the plants, whereas the Hox genes is to do with the body plan that is in the animals. So the head tail orientation. And then the question they once asked about homeobox, when they linked it to animal response, they actually linked it to animal response and asked you to talk about the fact that the Hox gene was involved in the development of the brain and the spinal cord. What about radial and bilateral symmetry? What do you mean? What about radial and bilateral symmetry? Radial symmetry just means that the symmetry is all the way around. So there are organisms like Canidera who have that. Why the fruit flies used for studying? Fruit flies are used for studying because they are very simple. They are very small and they reproduce really quickly. So that's the reason we use fruit flies. And bilateral symmetry is talking about the fact that you've got that bilateral symmetry on two. Bi means two lateral going down the symmetrical side of things. So my arms come out at the same point in that bilateral symmetry at the same point in the same position. And you'll find that with any type of, not that you guys have to go into that, but if you were looking at classification of specific organisms, you'd be looking at the fact that some of them are radial, some are bilateral, some are unilateral. But it's about where the mouth is, where the tail is, etc. Why would you want it to reproduce quickly? If you can get it to reproduce quickly, it means you can study lots of them and see what happens with them. So the homodomain, so this is where what you're doing when you talk about the homodomain and you're looking at, sorry, I keep watching the time because I have to, one, get snacks and also leave to go pick up my girls. So when we talk about the homodomain, we talk about the homeobox, what you're looking at is you're looking at the key definitions of what we're, so the part of the protein, the homeodomain that binds the DNA, switching the genes on and off, that is the homeodomain. So the homeobox gene are the groups of genes which contain the homeobox. The homeobox is a section of DNA, 180 base pairs. So that's what I was talking about before. So the homeobox is that section of DNA. When we talk about the part of a protein which is conserved, that is the homeodomain, and that is what binds the DNA, switching the genes on and off. So we therefore say that the homeobox genes are regulatory genes because they are the transcription factors which are going to involve that switching on or switching off of genes. And then obviously, if you're looking at the brain and spinal cord, just to finish this one off before I leave, if we're going to switch the genes on or off, we're there for differentiating the cells. So we're differentiating neural cells within the brain to be cerebrum, cerebellum, medulla oblongata, hypothalamus, pituitary gland, or are we differentiating cells to be whether they are your cells that are going to make up your phalanges, your carpals, your metacarpals, etc. So the Hox genes also regulate that level of apoptosis and mitotic division. And that's why as animals, we don't have the capacity of our cells to go through mitotic division after we get past the totipotent stage. They get to the pluripotent stage where they can become any cell type, but not a whole organism and so forth. So it's about linking back to your module 2. But guys, I am going to have to go because I really do need to ensure that I go pick up the girls. No, bilateral and radial symmetry is not in the OCR spec. No, it's not. I don't know. I think I'm going to work my way through based on my gut judgment of what I think is coming up. If I would give you any advice, I'd say go over biotechnology, and also make sure you're confident with the culturing microbes in fermenters. I feel like fermentation's coming up, but in the batch and continuous fermentation would be where I would think that would come up. Guys, thank you for joining me. I hope it is helpful. Obviously, I'm trying to do this as quick as I can for you, and having not had my lunch because I've just finished a client and gone straight to this. So you're very welcome. Keep watching my other videos because my other videos are actually more, I've planned them out, I've talked about them, they're more specific, whereas here I make sometimes some silly mistakes, and I'm human, just like you would make mistakes in your exam, but I hope you do find it useful. Anyway, guys, thank you.